Right, I mentioned that proteins that undergo post-translational membrane insertion, those are proteins that go to the nucleus, mitochondria, peroxisome, plastids, this like chloroplast. I don't really talk about chloroplast too much because I always assume it's plant people in the house. Are there plant people in the house? Okay, I don't mean to degrade you. I'll talk about chloroplast more if you are here. So in that case, those proteins, they move straight from the cytosol after being translated on free ribosomes to their destination. Proteins going anywhere else, their first stop is the ER. And really, their first stop is during synthesis on that ribosome. It becomes a bound ribosome, and they get inserted either into the ER membrane or into the ER lumen. And then from there, they can go to all these other places. And if that's the case, you're talking about vesicular transport. So we're going to talk a lot about how vesicles form and flow throughout the cell. And so this, when we're talking about a protein, so I found out the reason I kept trying to watch the videos on Firefox. Firefox doesn't really support YouTube anymore because it relies on Flash Player. So if you're trying to watch those videos, use Safari or Chrome as your browsers, in case you encounter that same issue. All right, so let's first talk about intravesicular or intracellular vesicular transport. So targeted specific transport of molecules. And again, it can be out of, into, or within the cell, mediated by membranes, right? And it's often referred to as the endomembrane system, so the membranes within the cell, endo-inner membrane system. I feel like that nomenclature is a little bit old. I explained why in the learning module. But it is what it is. So outward flow. So outward flow specifically refers to newly synthesized proteins that are translated on bound ribosomes. Okay, we'll get into why that is again. And then once they are translated on bound ribosomes into the ER, they rely upon trafficking in these membrane-bound vesicles as they leave the ER and move outward. So typically what happens is you start a translation on a bound ribosome. That's how you get in the door. That's how vesicular transport begins. If you don't get into the ER, you're not being transported by a newly synthesized peptide is not being transported by a vesicle. This is step one. So it gets into the ER, it gets some inlet glycosylation happening, maybe some lipid modification. It folds with the help of cholinesterase and calreticulin, and if it gets it right, it then leaves the ER in a vesicle, which is this tiny membrane-bound compartment that functions as basically like an intracellular car. And we're going to talk about how these vesicles form and move. So there's these types of proteins called coat proteins that help the vesicles bud. They kind of pouch out from the membrane. Transport is dictated by these motor binding proteins that bind to motor proteins that move along the cytoskeletal filaments that kind of function like tracks or railway tracks or like a road. The motor proteins move the vesicle along those tracks. And then you want to make sure that the vesicle goes to the right place, right? So if it's a lysosomal protein that's just been synthesized, you want to make sure it goes to the lysosome. So a vesicle is always packaged with an address, right? So if you're talking about a protein that's just leaving the ER though, the first stop is always going to be the Golgi. So proteins or vesicles that leave the ER, they go to the Golgi, and that's where processing gets finished. So there's all these glycosylating enzymes that will modify the way those oligosaccharides look. And there's some other modifications that also happen in the Golgi. Kind of think of it as, right, in the ER, the protein is being born, and it's getting dressed, but it's not quite done. Maybe it has its underpants on, but it's not ready to leave the house yet. So it goes to the Golgi and it gets the rest of its clothes on, and now it's ready to work. And so the Golgi then, from the Golgi, it releases a vesicle again, so the whole thing starts over, so you have to have budding, you have to have specific transport and targeting, and it gets packaged again with a particular address. Does it need to go to the plasma membrane? Does it need to go to the lysosome? There are specific addresses that the Golgi will help add to that vesicle, right? So it's very targeted. It's not just like, here's vesicle, good luck getting there. It's instructed on where to go. And we're also going to talk about how fusion occurs, right? So I already talked about how directed it is, right? The cytosolic side always fuses with the cytosolic side of the lipid bilayer, non-cytosolic. But there are proteins called snares that basically snare the two membranes together, and they pull the lipids so close that they can fuse. And we'll talk about how and why that is. Yes, sir.
Why would it go from the ER to the Golgi back to the ER until I make different protein segments?
There are a couple of reasons. A lot of times when the vesicles bud from the ER, there will be escapees. So like ER resident proteins that we're not supposed to leave, they're recognized. So there's a ER retention signal, 4 amino acids, KDEL, K to the K delta. So that gets recognized by retrieval proteins that are like, not so fast. They package them in a vesicle and send them back to their home. Another example is if that protein gets synthesized but needs extra modifications that only occur in the Golgi, it'll go through that modification process and get sent back to the ER. Great question. Inward flow, so if you guys have heard of endocytosis, that's when things move inward into a cell from the outside, so we'll talk about that in a slide that's just not all words, which are my least favorite types of slides. And then, again, membrane-contained movement within the cytosol. So let's look at some pictures. Right, so components of the endomembrane system were originally defined as either being directly connected, so like the nuclear envelope is directly connected with the ER membrane, or that exchanged material directly through vesicular transport, right? Vesicles bud from the ER and fuse with the Golgi. Vesicles bud from the Golgi and fuse with the plasma membrane, or fuse with a structure called endosomes, or fuse with lysosomes. And so that's how the endomembrane system was determined. And so, right, so you notice mitochondria didn't make the list, oxomes didn't make the cut, because for a long time they thought that vesicles didn't move from the ER to those structures. But surprise, surprise. I think that's exactly what I said in the learning module, surprise, surprise. There's now evidence that they do exchange vesicles, no one's gone back and redeveloped all the content to include them into the endomembrane system. Rant over, I don't know. So you guys don't have to memorize like, oh, endomembrane system means these things. I just want you to understand the concept of vesicular transport, right? How it's regulated, and the purpose of a vesicle. Like, why is it moving from the ER to the Golgi? How does that process happen? How is it regulated? The proteins involved in that. I.e., there's not going to be a question on the test, which one of these is the endomembrane? I hate those straight recall questions. I realize they're regarded as like the easiest questions, and so I try to throw you guys a bone and put those in there. And it's so funny, I have students all the time who, those are the ones they miss. Like they get the really hard conceptual ones, and then the easier ones that I hate anyways, because they're just straight, regurgitate, vomit back this information I gave you rather than think about it. Okay. So bridged by vesicles. All right. So the system is awesome because it allows for this really cool mechanism of directed transport to these different locations. And the other thing it does is it can provide a safe haven for like hydrophobic molecules that can't really make it in the cytosol, right? So if you have a hydrophobic molecule, it can be safely contained within the context of a vesicle. Or if you have dangerous materials, so like lysosome, right, is full of all these enzymes that break other things down, right? So the lysosome is like the stomach of the cell, it chews everything up. So you want those molecules to definitely be sent to the lysosome, right? You don't want them wreaking havoc on other parts of the cell. And so we have this cool system to allow neatly contained transport of those molecules to the lysosome. And then finally to get things secreted into the plasma membrane. You can just, instead of relying on a bunch of very specific transport proteins for all these different molecules that need to get out, you can put them into a vesicle, and cool, that whole vesicle can just fuse with the plasma membrane and dump them all out into the extracellular matrix. It's a really cool, efficient way to move things in a directed fashion through the cell. So inward flow, again, you have things moving from the outside of the cell in, they get endocytosed into a vesicle. There's 3 different flavors of that happening, and we'll go over those at the end of lecture. So outside in would be inward flow, and then autophagy. Autophagy, or autophagy, is self-eating, right? And so we have a lot of things that break down pretty quickly in a cell. Like the average lifespan of a mitochondria, I think, is only like 7 days, right? So the second things start to break down, we need a mechanism to get rid of them before like they lyse and wreak havoc in the cell. And so autophagy is a mechanism where essentially for materials that need to be broken down, a membrane will form around them and then deliver them to the lysosome by fusing with the lysosome. And this mechanism is important for normal cellular physiology. When it breaks down, it's associated with cancer. In 2016, one of the Nobel Prizes was awarded to Dr. Yoshinori Osumi. He was a scientist who years ago took the images that allowed us to really understand the molecular mechanisms of autophagy. Really cool. Really cool, and that's about all I'm going to talk about autophagy in this lecture. Definitely a phenomenal mechanism. Okay. So let's start with outward flow, right? And again, it starts in the nucleus ER complex. We have our newly synthesized polypeptide. Right, graphic illustrators always strip down images like crazy. What's missing from this image? What structure that should be here is not here? ER, yeah. So ER is all over the place in a cell, and some cells have more ER membranes than others. But here's an actual image of an ER. So here's the actual membranes, and right, the ER kind of lines back and forth, this big old membranous structure. And what I love about this image is it shows the black dots, these are all free ribosomes, and then the black dots here, those are bound ribosomes. And the ribosome, again, in its cartoon form, always looks like this, like, weird asymmetrical, like, hamburger bun with a large subunit and small subunit. And one phenomenon about the ribosome that I don't know that a lot of people appreciate until they come through molecular cell biology is that ribosomes are cycling between a free and a bound state. So ribosomes move back and forth from being bound to free. And we're going to talk about exactly how that happens. It's during synthesis. If the ribosome is translating a peptide that has what's called an ER signal sequence, it's recognized by a protein called SRP, signal recognition particle. It finds it, stops translation and moves that whole thing to the ER membrane. That binds it to the membrane. And then when it's done translating that peptide and releases it into the membrane or into the ER, it's released and recycled back into the pool of free ribosomal subunits to participate in more translation. Yeah.
The large subunits of ribosomes only attach to one small subunit?
At a time?
Yeah, what is it like one?
Oh, are they like polygamous? Can they? Yeah. So because the small subunit attaches first, and then it recruits a large subunit, and there's just a bunch of really similar looking small and large subunits. So you'll have small subunit attaches, and then once you get recruitment of all the EIFs and the start codon is recognized, then you get the large subunit. Once translation is done, they both release and go their separate ways to maybe find a new relationship with another subunit. It's kinky in there, man. I was in the curriculum committee last week, and someone from the history department came, and she was talking about all these cool history classes that she's trying to develop to embed like science-y stuff to kind of attract our students to maybe take history courses as their elective courses. And she was telling us a story that their dean told them to sex up all their titles. Like specifically sex up your titles. Like sex in something in history. And she's like, yeah, if you just put sex in the title, it's incredible how much our enrollment goes up. Makes sense. All right. So this image is, I love this image because, so this is an image of a whole cell, here's a nucleus, and check out that ER, right? Winding itself back and forth throughout the whole cell, right? The cell is a crowded space. It is not the way that, the computer, the CG imagers that create the graphics, it doesn't look exactly like that. It's messy, there's lots of stuff going on. And so this slide is just to remind you about what are those functions of the ER that you learned at some point and maybe have forgotten, or are important that you know now. So lipid biosynthesis, right? This is one of the primary generators of lipids in the cell. And so all types of lipids, right? We talk a lot about phospholipids in this class. There's a lot of other types of lipids that need to be produced in the cell, and they get made in the ER. So phospholipids, steroids, lipoproteins, those are some examples. So this next sentence, there is great cell specificity in lipid enzymes. So there are enzymes that need to be present to make all these different lipids, right? So when I say there's cell specificity in lipid enzymes, what do I mean?
Some cells produce more lipids?
What's that?
Some cells produce more lipids?
Not exactly.
Certain cells produce certain lipids?
Yes. So if you have two different cells producing different lipids, that means their ERs might have different lipid synthesizing enzymes, right? So like an example of that, liver, let's go with liver. Liver produces bile, right? Well, one of the components of bile are lipids. So the ER within the hepatocytes, so one of the parenchymal cell of the liver, if you will, it has the biliary lipid producing enzymes are enriched in the ER in those hepatocytes. But if a cell doesn't make biliary lipids, it doesn't need those enzymes. Right? All right. Membrane-bound translation of mRNA, right? That's one that hopefully we can all get on board with right away. So ribosomes that bind the ER, right? They participate in translation of mRNA. Those peptides move across the ER membrane. The way that happens is the peptide actually has an ER signal sequence. So the peptide itself tells the cell, I need to go to the ER, right? And so that is recognized by this other protein called SRP, signal recognition particle. So the ER signal sequence, it goes by different names. On a test, I will probably always call it the ER signal sequence because it tells you endoplasmic reticulum in the name. The ER signal sequence is sometimes also called N-terminal leader peptide. So right, if I did use N-terminal leader peptide, I would print in parentheses ER signal sequence. So the ER signal sequence is recognized by this protein called SRP. So what happens is it is oftentimes right at the N-terminus. Not always, but oftentimes. And so as that peptide is synthesized, the first thing that emanates from the ribosome is that ER signal sequence. SRP is a protein that's just hanging out in the cytosol. It binds to that sequence and helps transport the ribosome to the ER membrane. And it docks on a receptor that is on the ER called the SRP receptor. So all of those pieces need to be there in order to get a protein that is destined for the ER to the ER. To get a ribosome from free to bound state, it needs to be translating a peptide with the ER signal sequence. SRP has to be there to find the signal sequence, and then SRP needs to be recognized by the SRP receptor. And that's how we get it bound. And then as it's being translated, it's moving through that blue protein called the protein. What's the protein that translocates proteins through the membrane? Yeah, look at that, protein translocator. You guys are sleepy. You guys must not have had 4 coffees like Professor Mulligan asked today. Oh yeah, I did that. I did that. It feels good. Okay. The other thing that that happens in the ER, you get initial integration of lipids and proteins into the membrane. So if you have a transmembrane protein that's being translated onto the bound ribosome, it moves through the protein translocator, and that protein translocator is what initially embeds it into the membrane. It never leaves the membrane from there on out. It gets transferred within the membrane as it moves in a vesicle through the cell. So you get initial integration of that protein right there at the ER. Same thing with lipids. When the ER creates lipids, they're not just flying all over the place, they get embedded into the membrane. And if you need to deliver those lipids to another membrane, they move in the context of a vesicle, in the membrane of that vesicle. Okay. So the kinds of proteins that are made on bound ribosomes are going to be proteins that are destined for the extracellular space. Those need to move through vesicles to get there. They are proteins that are destined for the plasma membrane. Right? So any proteins that function as extracellular signaling receptors, they are going to need to be translated on a bound ribosome to get into the whole vesicular transport system. They need to get in that door so that they can be transported via a vesicle to get to the plasma membrane. No other way that they can get that. Or proteins that are destined for lysosome, Golgi. All of these proteins are going to have signal sequences that instruct delivery to the ER. Every single one of these are going to have ER signal sequences, right? That is how they get into the starting point for vesicular transport. And so once they get into the ER, the next step is getting trafficked in vesicles to the Golgi and then out to their final destination. So in contrast, the proteins that are bound on or made on free ribosomes, so these do not have an ER signal sequence, are going to be proteins that they're just going to work in the cytosol. So why would you put them through the ER? There's no purpose. Just translate them in the cytosol, and they're there where they need to work, right? So, so here are examples that would have made a lot of sense to you if we'd already gone over the cytoskeleton lecture. So this lecture used to fall after cytoskeleton. What are some cytosolic proteins that we've talked about in class that I'm going to ask you about on the test? I'm going to think of 2 that start with the letter H. Yeah, look at you. Yeah, HSP60, HSP70. What's a cytosolic protein involved in translation? Yes, you got it. Yeah. So those would be examples of proteins that would be translated on free ribosomes. Proteins that need to live in the nucleus they're going to be translated on free ribosomes and then be transported via proteins directly to the nucleus, and then mitochondria, free ribosomes, and then going to go into the plant people chloroplasts as well, and then also peroxisomes. All of those also have signal sequences. It's just instead of them causing the ribosome to be bound, they don't cause the ribosome to be bound. What they do is, after synthesis, those signal sequences function as the site where the transport protein binds and takes them to their final destination. Oh, good, fantastic. Okay. All right. So important to know this. So signal sequences. I just want to. I started including this slide on signal sequences to help provide more clarity on them, and also to contrast them with signal patches, which is a concept that students had a harder time with before I had imagery. When I just tried to like hand wave, they were like, huh? And I was like, what? Huh. Okay, so a signal sequence, which is often used interchangeably with signal peptide. Okay, so I will use them interchangeably, and just know those are the same thing. Signal sequence, signal peptide, it refers to this contiguous stretch of amino acids, right? So the signal peptide is drawn in red. So the amino acids that make up that signal peptide, they're all right next to each other in the sequence. Okay? And they help indicate to the cell where they need to go. In the case of the ER signal peptide, the way they do that is by binding directly to SRP. And then SRP binds them to the SRP receptor. If it is a nuclear protein, it has a sequence called the nuclear localization sequence that binds directly to these factors that know to deliver that protein to the nuclear pore. So that is different than a signal patch. A signal patch only shows up post-tertiary folding. So a signal patch is composed of stretches of amino acids that can be really far apart from each other if you stretch out the peptide from N-terminus to C-terminus. And they only come together to form that signal patch after that protein is finished folding. And we're going to talk about a very specific signal patch when we talk about how proteins make it to the lysosome. So proteins making it to the lysosome is at kind of towards the end of the structure. And it requires a signal patch. That is the first indicator. It has a specific signal patch that is recognized in the Golgi and it helps a tagging mechanism occur that tags it right away. It says, be careful, cell, this needs to go to the lysosome. So let's take a look at how the ER signaling sequence works. This is an image that's actually on the learning module. So it helps provide, I think, a little bit of, hopefully, clarity on how this happens. So again, a signal peptide or a signal sequence, it's short. They range from typically 5 to 30 amino acids that functions as an address, right? It helps determine the eventual location of a protein in the cell. And they bind to a dedicated transport protein. So in this example, the transport protein is SRP. It transports the peptide along with the ribosome and the whole kind of translation complex to the ER. So they're most commonly found at the N-terminus of proteins, but there are examples of them being at the C-terminus or being internal. So that's not like a for certain rule. For a long time they thought it was, and then most things, science was offended again. What? Science controversy. Alright, so here is our ribosome that is staying on the cytosol, right? And here's the RNA that's being translated, and here's our what's always referred to as a nascent peptide. Nascent, someone tell me what nascent means, because that you might see that word on the text. That's a term that's used all the time in science. Yeah, newly born, right? So think of it as like the baby head just emerging. So it's newly born, but it's not finished being born. I'll spare you from the birth story that I just thought about telling you. Do you guys want to know how long my delivery was with Emmett? Yeah.
24 hours.
38.
What?
50. What? 50 hours. Yeah, it was a marathon, and of course I was like, no, as a biochemist, I want to feel all the feelings that we feel. And so I was like, no drugs. And by the end, I was like, why did I do that? But I also felt like a superhero. I was like, I did it. Yeah, and he was a 4th of July baby. So he was born 7 seconds to midnight. And so it was 10 minutes to go, right? And it was cool because he was born at UCSF. And it's on this hill, and so we had these cool, the birthing center is on the top floor, so we had these amazing views of fireworks shows that everyone else was paying attention to, but me, I had like an idea that there was changing colors out there, but right, I'd been awake for 72 hours, because my water broke at 9 p.m. at night, and so I'd been awake that whole day already, right, and so, I think 72 hours is off, but I was up for a really long time, and then, so then I was pushing for 2 hours, and I was so tired, and it was 10 minutes to midnight, and this awesome nurse comes over, and she was like, girl, do you want a 4th of July baby or a 5th of July? I was like, 4th of July! I was like, you better bear down, honey. I was like, okay. And my sister was standing on the other side of the door. I told her not to do that, and she was like, you made sounds that no person could make. She still does not have kids. That might have affected it. But yeah, and then of course he was born. It was so amazing, and then Ellie, guess how long it took her to be born? 3! We barely made it to the hospital. We got there and I had a doula, which is like basically someone who kind of helps with the birth process, and she was like, do you want to walk the hall? I was like, no! Yeah, she was just like, I'm ready, Mom. Whereas Emmett was like, maybe I do like my home and here a little more. How the heck? Oh yeah, nascent. So the head is being born, and it's kept jumping by the nose. The nascent peptide is emerging, and what happens is, right, that sequence is right there. It's right there for SRP to go, aha, I've got you. And so what happens is SRP grabs it, and it's this big old protein that grabs the whole ribosome, and translation actually stalls. It stops translation and then docks it right on the ER membrane. SRP binds to a receptor that's conveniently named SRP receptor. So if SRP receptor wasn't there, do you think this would dock to the membrane? Nope. Critical piece. You need all these things for the ribosome to bind. So what happens next, right? SRP receptor binds SRP. That binding, right? Protein-protein interactions causes conformational changes. It causes a conformational shift in the structure of SRP so that it can let go, but its work is already done because it nicely passes off the whole complex to the protein translocator, which this first signal, the ER signal sequence, it also functions as that initial start-stop transfer sequence. So protein translocator is like, oh, I got you, friend. And it starts translocating it through into the ER lumen. Okay, good? These stories I tell, they're actually strategic because I always see what happens is when I tell them, all of a sudden everyone's like, oh, I'm awake again, and I'm paying attention. I'm like, right, telling some story about poo or whatever, and then all of a sudden I'm like, they're paying attention. Back to molecular biology. It's good to have mental breaks, right? Okay. Brain can only take so okay. So now we're getting into, once we have this protein into the ER lumen, it's going to go somewhere else, right? So how does it get there? It relies on this vesicle that forms from the ER. And so this image, here's our ER, and you see these vesicles forming. So that's what they actually look like under a microscope. And a lot of times the vesicles form to form what they refer to as a vesicular tubular cluster. I don't you will never see the word vesicular tubular cluster on a test. I'm just talking about vesicles. So that's that's next level. So the way they bud off from a membrane is with the help of these class of proteins called coproteins. They basically coat a small region of the ER membrane, and they form this cool lattice-like structure that reminds me of like the jungle gyms at parks. And the way they form, this lattice just basically pulls the membrane out into a bud. So coproteins do budding. The vesicles, after they form this bud, the coproteins dismantle, and then motor proteins will connect to something called a vesicle motor binding protein. The protein that gets recruited to the surface of the vesicle that links right up to its wheels, the motor protein. So motor proteins are these class of molecules in cells that do all the transport. There's different types of motor proteins. They always have these heads that can walk along cytoskeletal filaments. And so it's not like vesicles are like swimming freely, like bubbles or something. They're connected to a motor protein that in a very directed way, walks along cytoskeletal filaments and takes that vesicle to its destination. And then targeting, right? I talked about how vesicles, it's not like they're just sending them off and saying like, all right, hope you get to the right destination. There's a protein that gets coated on the surface of the vesicle called a RAB. And there are all different types of RABs. They have numbers, right? So it's like RAB1, RAB2, RAB3, RAB4, RAB5. And each RAB will target a specific destination membrane. Right? So say I don't have these memorized and I don't want you to memorize them either, but say it gets targeted or it gets RAB7 gets added. Well, maybe RAB7 goes directly to the lysosome. So RABs dictate targeting. And then finally, there's fusion. And fusion, again, it's going to be determined by the lipid constituents. So again, that's getting back to what I drew, the outer leaflet and the inner leaflet. And then this protein called a snare, right, which snares the vesicle membrane to the target membrane and pulls those lipid bilayers so close to one another that they can fuse. So those are the details. And again, it's this picture again, right? So coproteins help do budding. Transport is motor binding proteins, which bind motor proteins. And then targeting by RAS. And then the membrane proteins called snares, which help tether them together. So 414. Oh, okay. Last thing. So remember, once cytosolic, always cytosolic. Once non-cytosolic, always non-cytosolic, right? So I colored this purple so you could follow it. Really make sure you understand why that is. Okay, so we'll